In the course of next half an hour, 35 minutes to 40 minutes, I will be talking about our works also in the context of the paradigms related to evolution of bacterial pathogens and how they adapt to the host mounted immune system and as well as the environmental pressures. So together, the environment and the host, they are crucial to shape the pathogen to evolve their arsenal of virulence and other fitness related traits so that the most smart lineages could emerge over a period of time during the evolution. So it is quite interesting also for me as it is part of my career track of work. And this is also in the mainstream because of certain emerging problems such as the antimicrobial drug resistance and emerging and re-emerging infections in different domains such as public health, animal health, environmental health, food safety, and so on and so forth. I hope I am sufficiently audible to all of you. If there is any problem, you can just ask me. And I hope the slides are also transitioning. So as you can see from here, the paradigm of the evolution of two different classes of pathogens. They are both bacterial pathogens, mycobacteria and enteric bacteria, as exemplified by the E. coli here. On one hand, mycobacteria, they evolve through shedding their genome. So genome miniaturization or minimalism of the genome is the predominant strategy that the mycobacteria adopt to evolve fitness advantage or the emergence of specialist lineages, as we call them. Why they are specialist is that each of these lineages are then tailored to a particular destination host. For example, Mycobacterium tuberculosis is for humans. Bovis is for the cattle. And Mycobacterium microti is for the voles and rodents. Mycobacterium caprae is for the goats. Mycobacterium pinnipidae is for seals. So you can see the callout here on the left-hand side bottom panel. You see that each time mycobacteria have resorted to downgrade their genome at a specific coordinates, thereby losing chunks of their genome, they have tended to evolve as a specialist lineage which is destined to a particular host species. So that paradigm we could easily observe when we align the genomes of different mycobacteria and see which of the chunks of the genomes were lost and what is the timeline of their losing these genomes over a period of time. So that is, in short, is the modus operandi that they have adopted over a period of time to evolve into the fitness advantages of their kind so that they can tailor themselves to particular host species. Gram-negative bacteria, on the other hand, as also exemplified by E. coli, Helicobacter, and other pathogenic organisms of the gram-negative clan, they have been evolving on the contrary through aggregation of the genome. That means they are acquiring new genes, new genomic regions through horizontal gene transfer. So these new genomes or the genetic elements that arrive in their gene pool could be contributed by plasmids or the phages or the pathogenicity islands, genomic islands, and so on and so forth. Owing to their high propensity to integrate foreign DNA in their own DNA, or their high capacity or their propensity to undergo genetic recombination, they have been traditionally evolving through horizontal gene transfer or lateral acquisition of the genome. In both the strategies, the model, the purpose is to evolve the fitness, the optimization of the metabolic repertoire, optimization of the survival mechanisms, and optimization also to inflict upon hosts a balanced cost so that the host is unable to clear them out and they are able to sustain and survive at the cost of the pathways encoded by the host machinery. So that is how we have been seeing these evolutions as two contrasting paradigms, which we have also reviewed in a fairly nice review written in 2008 for Nature Reviews Microbiology. That was an invited review article that we have written. which you can read. The reference is there. And there we have highlighted all these things that I have just discussed. And this is also visible at population level. It's not just the theory of it. When you analyze the genomes of bacteria and they see their fingerprints at population level, and population level, I mean the thousands of genomes when you fingerprint from different parts of the world, and when you see, for example, gram positive, the gram negative bacteria and mycobacteria, how they look like on a two-dimensional gel. And so you will see that while mycobacteria appear to be highly clonal and similar, what I mean is that each fingerprint of a single isolate looks very similar to the other isolate. That means they have been shedding so much of their genome that much of their genome has now remained to be only clonal. Like a lot of flexible part of the genome has already been discarded, shedded, or deleted, or has been pseudogenized and eventually discarded. So what is required bare minimum for mycobacteria to survive and remain as a pathogen over a period of time is there. And that is how the fingerprints are so clonal and so similar. Whereas in case of E. coli or Helicobacter, since they are aggregating their genomes over a period of time, they are resorting to horizontal gene transfer. Their fingerprints are very diverse. So that is what is the message here. Like these paradigms of genome evolution are also visible when we genotype them and fingerprint over a period of time or on a population scale. So such that by just seeing the gel image, you can make out that which one is clonal, which one is the diverse population. When we put forward, on a landscape of their evolution where a common ancestor of mycobacteria has been having a large genome, the deletions are such big deletions that once they are lost in evolution, they cannot be gained back. So reversion is not possible. That is how this is only one direction evolution that is happening. So they are deleting their genomes and therefore they are evolving from most flexible type of bacteria to most restricted or parasitic kind of bacteria. So for example, the ancestor of mycobacteria here in the cartoon that I am displaying in front of you, the ancestor could have been very flexible, having lots of pathways of independent survival other than those genes that may be required for virulence. From that ancestor, which was like having 8 or 9 MB genome, they have successively shedded their genes and chunks of the genomes over a period of time, and they have evolved into sort of a very highly parasitic, quote unquote parasitic. I mean, how a bacterium can be parasitic is that it doesn't have its own metabolic machinery intact. So for most of its metabolism also, it is dependent on the host. So Mycobacterium leprae, for example, its genome is, little less than 3 MB. So this Mycobacterium leprae has evolved at the cost of shedding close to 6 MB of the genome over a period of time. And this time scale is like millennium, 3 million years. That is the timeline. And that is how, the most smart and parasitic and ruthless kind of mycobacteria, such as Mycobacterium leprae and Mycobacterium tuberculosis, they have come to existence through this evolution by deletion over a millennial scale. Okay, so this paradigm you see here, keep in mind that the ancestor may be very flexible also as regard to host tropism. So the ancestral bacteria may be able to infect many different type of animals and humans, whereas the most parasitic type of forms have restricted themselves predominantly to the human host. Okay, that is how the mycobacteria have been evolving. Helicobacter also has been similarly evolving, like their ancestor could have been through this paradigm which I am displaying right now, could have been a non-virulent, a non-pathogenic commensal type of organism. And over a period of time, we have juxtaposed it to the arrival of agriculture and how the agricultural practices have continent-wise, they have evolved, and how the Helicobacter populations have evolved in a continent-specific manner. And then they have, in a geographical specific manner, they have adopted themselves. Say, for example, the Helicobacter that have adopted themselves to Europeans is not able to survive in the Japanese stomach, and vice versa. So they have very nicely fine-tuned themselves to the host populations, such that the Japanese Helicobacters could only survive in the Japanese stomachs, and so on and so forth. So this geographical evolution of Helicobacter is also very, very dramatic. And also when we juxtapose it to certain things that have also arrived in a geographical fashion, such as evolution of agriculture. So we get this kind of a scenario whereby it is tantamount, or it appears that some of the microorganisms which were present in early cultivated plants or domestic animals could have donated certain virulence apparatuses to Helicobacter, and through horizontal gene transfer, it would have acquired that and that level of fitness in order to spread in a geographical manner. Okay, so with this background, we move forward and we see what is the status of today's smart superbugs. While their ancestors have been like this, so and then they have been evolving. So it is very clear that they evolve in tandem and evolve in a specific manner, also with respect to as juxtaposed to their host as well as the environment. So both the host and environment are very important in their evolution. So when we discuss this in the context of the present day problems such as the antimicrobial resistance and the resistance traits that drive their fitness into the populations, we see that predominantly two different forces are working on the evolution of these organisms. They are the bacterial genetic driven forces and the environmentally driven forces. Okay, environment could be the host environment itself, like the gut environment, or like the, for example, the exterior environment, it is possible. So wherever they go, if they go into the gut, they go into the exterior environment, whatever is the vicinity is their environment. So there are host of factors, for example, that shape the genome evolution in these bacteria, also with respect to their resistome. So resistome is the collection of resistance genes. So many different genes that encode or that facilitate or support the shaping of a resistance phenotype. That resistance phenotype also loosely we call it as a fitness advantage. Like resistance is gained by the microorganism to augment its fitness. Okay, so fitness cost is equivalent to their degree of resistance. So in the rough terms, you may argue in academic terms, the different definitions are there. But to convey, in the most lucid terms is that there are different, driving forces that are shaping the repertoire of these resistance genes and the resistome thereby, and also how they are integrated and functionally adopted in the larger scenario of the bacterial gene pool. And so predominantly, I see the two different forces, bacterial genetic forces and the environmentally driven forces. So selection during transmission, and the point mutations, horizontal gene transfer mediated acquisition of plasmids and mobile genetic elements, and so on and so forth. So these are some of the things that are from the bacterial side. And from the environmental side, the climatic and ecological forces and anthropological activities, like the human societies related actions, like for example, the pollution and human living, that is also having an impact on the evolution of these organisms in the close vicinity of the human societies. Soil, water composition, then other things, they all contribute to the development of these MDR or multi-drug resistant clones or the superbugs. So directly we have come to the present day microorganisms and what are the associated problems. Why there is a relevance of the evolution
Hi, Dr. Niyaz.
Yes.
There's a question in the audience. Yeah.
So are the questions to be right away asked in real time, or they are to be
whatever way you
of the talk?
If you prefer, we can take all of them.
Maybe one or two I can take, if they are required to take to understand the rest of the lecture. No problem. Go ahead, please.
Yeah, maybe we'll keep it to the end then, Dr. Niyaz. Yeah.
That would be fine.
Okay. Thank you.
Unless it is very important to understand the whole of the lecture, then we will. So what I was telling is that the evolution of bacterial pathogens that are rampant in human communities and their surroundings, and they are gaining the multiple drug resistance is important to understand as regard to the factors that which contribute to evolution of these organisms or microorganisms. And some of these factors are anthropogenic factors, as we have seen in this slide, and we will dissect them a little bit. For example, some time back, there was a front page cartoon in the Nature magazine, and in that it was written, "Superbugs are rising." And in the backdrop of it was the photo of a shantytown. So I was like wondering, what is the relevance of projecting a shantytown in the context of MDR bacteria? But then we saw the granulated data, in the context of our own country, if not the African countries and other South Asian countries. So some of the important things that are important, that are significant in understanding this scenario of evolution is like the poverty, slum dwelling, overcrowding, poor sanitation, poor management of drinking water and excreta. These are some of the indicators that are very important that have a direct bearing in shaping the resistome. I mean, the repertoire of the resistance encoding genes. So how much is the slum dwelling population, whether the closed drainage or open drainage or no drainage systems are there, and like that. So these are some of the data are from the census department, and they are inspired by the 2011 census. And the recent data are still not available. So we hope that during these 10-15 years of our development, that we may have seen, like, we call ourselves as, I don't know which era, that we smart cities era, or smart development, or Vishwa Guru, or whatever we have become. So whether these indicators have changed over a period of time, that will only be eminent when we get the data, latest data. So how the situation has been ameliorated. Okay. So we'll see whether we have progressed to the Amrit Kal or we are still jostling with the ground realities. Then we come back again to the ancestral evolution of bacteria with this background, seeing the different lineages that we see in mycobacteria. So when we dissect the population of mycobacteria, for example, mycobacteria circulating in this country, in India. So what we did long ago with together with Pasteur Institute in Paris, we had a program and whereby we took isolate from all over India and then sequenced them, genotyped them carefully, and also with respect to some of the polymorphisms and direct repeat regions. When we analyze collectively, we saw that there are certain lineages which are predominant in the Indian isolates of mycobacteria, and we call them as ancestral isolates or ancestral lineages. And we found that the ancestral Indian lineages are somewhere around 40% in India. And then on the contrary, there are modern lineages such as Delhi type and the Beijing type and other types like East African, Mediterranean, and so on and so forth. They are very modern, like they have high propensity to evolve into drug-resistant forms, whereas the ancestral strains may not be having that much high propensity to evolve into the drug resistance forms. So thereby the ancestral could be amenable or they could be manageable as regard to treatment and so on. And the moderns could be, refractory to treatment, if I put it that way. So this is the broadest scenario of the mycobacterial population circulating in India. And this is based on a modest collection of isolates that was analyzed in 2006. And since mycobacteria are highly clonal in nature and their genomes are rarely evolved very dramatically as gram-negatives evolve, so this situation is more or less same even today, because we do not experience a lot of population migration as the Western countries they experience or other countries they experience. So we are more or less a confined population, although we are very dynamic, a lot of, mobility is there, a lot of transportation, but it is all within the country. So with that, the scenario is roughly like this: 40 to 50% of the ancestral lineages, the rest of the other lineages. So if there are more ancestral lineages circulating into the TB landscape in India, that is a good news also for the control programs, the national TB control programs, that these lineages or these strains could be manageable. They may not be refractory to treatment if the treatment is, judiciously followed. So that is the meaning. Otherwise, in all other countries where a lot of modern strains are circulating, for example, some of the states of the former USSR, the TB is still, TB strains are still not manageable, and a lot of drug resistance is still prevailing. So that was the message of this study that we did. Then, there is another factor that can be integrated into evolution of bacteria, which is climate change. So now I'm going towards the gram negatives, which are very highly influenced by the environmental factors, where the mycobacteria are not that much, influenced by the environmental factors, because they are basically host restricted pathogens. I mean, they are inside the host, deeply embedded in the calcified nodules, in the, lesions of the lungs. So they rarely see the environment, and of course they cannot survive in the exterior environment, very sensitive to sunlight, very sensitive to the dry and humid environment, so they may not survive. So the environmental parameters, climate change and all may not be very relevant in the evolution of mycobacteria, maybe from the point of view of overcrowding and those kind of things, which may help enhance transmission, but it is very relevant in case of the evolution of gram negative bacteria. For example, the climate change and human migration over a period of time are the pivotal drivers in some of the resource poor settings. The Himalayan glaciers are melting, and therefore the sea levels are rising in the Bay of Bengal. There was a convention published in Scientific American few years ago, which put forth that each time the Bay of Bengal gets into the peninsula by 1 feet, 1 million people are rendered homeless. That is the rough convention. So that means the rising sea levels have a huge toll on the agriculture, the people's livelihoods. For example, if there's more salinity in water, there will be hypertension related issues, difficult births and deliveries. We call it as preeclampsia, rise of preeclampsia. The lands will turn barren. People do not have clean drinking water. So these conditions force the people to migrate. And then the human migration, when it happens, when they arrive in the cities, so these people create either the new slums or overpopulate the existing slums. So there is a burden of the drinking water management system, the sewage handling system, the garbage collection system. So in some slums, these things are non-existent. So the situation is overflowing. And because of that, the microorganisms get an opportunity of the gram-negative type to evolve, to transmit, and to, propagate themselves to the great scales. That is the meaning of it. And you see the picture on your screen. These are some of the conditions where you have a lot of filth and you have a scarce supply of the drinking water, and the people are living there. So this is how the concept of urban slum health is very important. And on every square inch, there are different biofilms, different microorganisms with varied virulence potentials and different capacities to survive and inflict upon the host pathogens, the host pathogen dynamics. So these are to be studied. These are some of the areas that are very dear to our heart, and we would like to pursue them over a period of time through genomic analysis of the resistome and through the associated factors, how the resistome is functional in the larger realm of the gene pool of a particular species, how it is changing over a period of time, what outbreaks and what type of diseases and symptoms are being reported, whether it has a impact on the growth of the children and other communities, whether they have any impact on the developmental milestones and other socio-economic milestones and indicators. For example, the attendance in the classrooms of the school-going children, whether that attendance is affected by some of the infectious episodes. And if we intervene with the hygienic practices, whether the attendance can be brought up, or whether people can have good body weight gain and those kind of things. If the certain hygienic measures are, for example, the hand washing with soap solutions, clean drinking water. And sometimes these interventions work like a vaccine or work like a therapeutic. When the vaccines cannot be feasible, the therapeutics cannot be feasible, these things at least appear to be feasible. So from this thing in between, I will bring also the socio-economic, environmental indicators and factors for you to understand how these bacteria have been actually evolving. For example, the case of cholera is very relevant in case of such slum populations, slum dwelling populations, because the cholera is essentially a waterborne disease. It's a gram-negative pathogen, which has a very fast life cycle. It divides very fast, and it overwhelms other bacteria in the gut in, for example, a 24 hours period of time. But cholera also, bacteria also overwhelmed by certain phages in the environment. So the phages keep track, control through the quorum sensing mechanisms. The bacteria sense the preponderances of the phages, and when the phage populations go down, then these kind of bacterial populations, they flourish, and they have the reservoirs. I see in your slide that Vibrio cholerae has so many reservoirs in the environment. They could be the microplankton, the zooplankton, for example, in the estuaries, in the sea, in the rivers, river beds. And that is how this infection in certain countries, such as in Bangladesh, for example, in Odisha, in our country, and in West Bengal, some of the cholera episodes are seasonal in nature. Whereas rest of the country, the episodes of these infections are actually accidental. For example, food poisoning, this and that. But then in for example, the countries that are adjoining the Bengal basin, this is a seasonal occurrence. And you will see that in terms of incidence, there's a seasonal bimodal occurrence of cholera every year. And this cholera bacteria evolution is affected by many factors. As I told you earlier, the human population dynamics, the prevalence of the phage populations, then whether or not the microplankton or phytoplankton bloom is there or not, or the ecological factors such as the river discharge and meteorological data such as the rainfall and those kind of things. So it is possible to model the epidemic of cholera, and predict whether the cholera will strike this year or not based on all these factors. So there has been also an effort to integrate all these factors together with the genetic factors in order to evolve a dynamic algorithm whereby you can just advise the health enforcing agencies, whether or not these kind of infections occur and to what severity and what preparedness will be required. So this and other works that we have published, you can see our published work on Vibrio cholerae genomics and its ecological implications also in terms of prediction of cholera and so on and so forth. Another gram-negative bacteria of the similar magnitude, quite dreadful than cholera. Cholera can be treated with just simple rehydration. Like if you rehydrate and administer the saline solution, there is no need of administering any other drug. The patient recovers in 2.5 hours. But salmonella is very dreadful in the sense that the patients may not recover. Sometimes the antibiotics may not work because of the rampant drug resistance. And cholera also, just like Vibrio cholerae, has a huge interaction network. If you see the host populations where salmonella bacteria survives, anything that it can interact with, the large warm-blooded animals to, the primitive cold-blooded animals, the planktons, the aquatic plants, the protozoa, and, fishes, shellfishes, phages, anything, leeches, serpents, anything that you'll find the salmonella to be surviving, to be invading, actively dividing, or using them as a intermediate host or so, or like that. So salmonella work we are right now continuing on the same lines as to whether there are any socio-economical, ecological, climate-related drivers, and whether salmonella bacteria can be similarly, epidemiologically analyzed and predicted and forecasted. But difficulty here is that Vibrio cholerae is just one species. And it is a host-restricted and human-restricted pathogen, although it has a reservoir in different animals, but it is predominantly human-restricted pathogen. But in salmonella, on the other hand, there are so many species. There are many different serovars. There are many different host and destination species, and therefore it is very complex, the situation, and it cannot be justly equated with the paradigm of Vibrio cholerae. But overall, in a gram-negative bacterial scenario, they all fit very nicely. When they are evolving in the backdrop of environment and climate change, what is happening in the clinics is also important. When we see the antibiotic resistance that is reported in the clinics, for example, here the data I am showing you from Dhaka city, I had an opportunity to work there in an international position, and we saw that over a period of, for example, 12 years, or 12, 13 years, we saw that antibiotic resistance has gone up to all classes of antibiotics. 14 classes of antibiotics. Some of the organisms are 100% resistant to all antibiotics. We have one bacterial organism in our lab. We use it as a reference strain. We have also genome sequence published it as a taxonomical study. We call it as NA114. It's E. coli. It is resistant to 14 different classes of antibiotics. So a superbug of its kind. But not only that, this bacterium is available with us. Now we see that many of such bacteria are already there in the environment and they are resistant to all available antibiotics. When we see them in the clinic, you see the scenario here that all antibiotics are showing resistance. But what we saw is that chloramphenicol, against chloramphenicol, we saw that the resistance has come down over a period of time. You see the last panel in the slide below. Cotrimoxazole and chloramphenicol. So what could be the reason? The reason is that these antibiotics have gone out of fashion. So not many people are prescribing them. So present day bacteria may have not seen these antibiotics at least for a decade, and that is how they appear to be sensitive. So there is a possibility, like for these data are based on a lot of records that have been analyzed. You say, if not millions, maybe hundreds of thousands. And based on that empirical, whatever data, granulated data we have in hand, it is possible that some of the antibiotics like chloramphenicol could be brought back. But then who will convince the pharmaceutical manufacturers to start making them? It may not be profitable for them to go back and manufacture it. Otherwise, this, doctors say that chloramphenicol is a very good drug against typhoid bacteria. So you see the scenario of evolution, the scenario in the field and the human societies, and then the scenario in the clinics where the drug resistance is reported. It is not that only the municipal settings or the medical facilities report that. In the refugee setting, for example, we had an opportunity to work with UNICEF in the Rohingya refugee camps in Bangladesh. So Ukhia and Cox's Bazar, these are some of the districts which are providing a refuge to these forcibly displaced communities from Myanmar, which are like 1 million people living in a 3 or 4 square kilometer area. So very high density setting. And here we observed that in this study that we have published also in Frontiers in Public Health and in Gut Pathogens, we found that the ESBL producing E. coli were isolated from drinking water of the hand pumps. 6,000 plus hand pumps have been installed by the UNICEF in these Rohingya camps. And when we took the samples from the hand pump water, we found rampant E. coli and fecal coliforms there. And at the point of use also in the household where they store the water, from that sample also a replete number of E. coli and fecal coliforms were isolated. And not only that, they were simple fecal coliforms, but they were multi-drug resistant superbugs. So when such a controlled setting fails, to deliver safe drinking water to communities, then can you imagine in uncontrolled and, informal settings, these are funded by international agencies, for example, and international crisis it is, people are monitoring this crisis for several years. This is a political solution. Then under such a strong watch also, you can see that the infection is flourishing. There is no hygiene, and therefore there is a possibility that the epidemics might rise, and these people might suffer. The bottom line is that at least they cannot keep a good health. Even if they suffer, do not suffer with, institutionalized outbreaks or diseases. The bottom line could be, or a minimum that you can expect is that their health indicators will be definitely compromised, because these microorganisms will colonize the gut, they will colonize the soil and form the biofilms also in the utensils and everywhere. And therefore, it is very important to introduce the hygienic practices, make aware also the implementing agencies, the developmental agencies, the development foundations, UN international organizations. And that is how some of these studies are also targeted with that ambition or with that purpose, that people will take these issues seriously, that infection burden in communities is really a serious issue. And embedded in that is the evolutionary dynamics of these bacteria to survive, because they are very recalcitrant. They are very, dynamic. So their survival is very, very effective. So to break that survival chain is very important. And how we can break, whether antibiotics is the only solution, or disinfectants are the only solution, or whether there could be any other intervention mechanisms. So I have given you the scenario in the field, then in the clinic, then in the refugee setting. I mean, these kind of works are also very important because they have something to do with the human development indicators. And I will highlight that the institute where I was working, in 2016 onwards, 2017, we got the Conrad N. Hilton Humanitarian Prize. So this prize gives $2 million US, as a prize money to the institute and the working groups. And whereas the Nobel Peace Prize gives only $1 million. So that is very important. But this is definitely lesser than the Nobel Peace Prize. But in terms of monetary value, it gives more money. So that the field programs can be taken up and more change can be brought into the life of the people in real time, for example, through different interventions, the drinking water sanitization, hand sanitation, hygiene, separate management of the excreta, and so on and so forth. But the excreta has to be managed properly. If it is not managed and if it is mixing with the drinking water, what you can see is the world over, the scenario is not improving. It's not the question of only Nepal, Sri Lanka, Bangladesh, India, or any of the South Asian countries, but all over African countries, some of the South American countries, when the data were analyzed from the sewage samples on a global scale as to how is the AMR dynamics as revealed from metagenomics of the sewage samples from different communities, the message that has come is that the gene diversity and abundance, they vary by the region. If the region is socio-economically very underdeveloped region, there is a high abundance of antimicrobial resistance genes of critical nature. Okay. So therefore, the sanitization health improvement could potentially limit the global burden of AMR. So these kind of studies give us the indication that how much influence of hygiene and sanitation, and mismanagement or poor management of human waste and excreta have on the development of the resistome at the global population scale, and whether interventions, if provided in these areas, could improve that situation. I hope it is clear to you. We are going now from less complex to more complex studies, and at the end we see how we will conclude. But you will have an opportunity to ask questions, so just keep your burning questions, but also give me the feedback as to how we can improve these things. In Bangladesh, for example, the similar studies that I am telling you, this is the global study from all over the world. The samples were taken of the sewage samples. But what happens to the water samples? Forget about the sewage samples. Even the surface water samples that were analyzed had they were particularly very abundant in antibiotic resistance genes. The urban waters, surface waters, or the rural area surface waters are having high level of antibiotic resistance encoding genes, or the AMR genes. So even the freshwater systems are not spared. And this is, is the direct surrogate of overcrowding, the population migration, the poor living standards, poor sanitation and hygiene, and poor segregation of farm animal communities and human communities, excreta, and those kind of things. So you see that this was a M Systems paper from Bangladesh, and here also you see that the urban and rural sediment and water had a lot of repertoire of resistome of these gram-negative bacteria. Over a period of time, so because of this, why? in 2010, 2019, people were observing that steadily there is an increase of antibiotic resistance. So underlying reason for that is that the resistome is shaping in real time in a dynamic fashion, and it's ever increasing. So we see antibiotic resistance as a function of resistome reshaping, the resistome dynamic spiraling, the resistome expansion, and some people see antibiotic resistance as a patient non-compliance, as a inefficient and counterfeit or duplicate drugs that are being made available in the market, or the the doctors are not able to understand and or prescribing, or over prescribing or under prescribing certain antibiotics, or giving broad spectrum antibiotics for a narrow spectrum problems. So clinically there is a different viewpoint, but from a bacterial genetic point of view, we see it as a direct function of the spiraling, resistome dynamics, because of the interaction of gram negatives largely with the environment and the host. See, cholera is rarely treated with antibiotics, as I told you before, but you still see that there is an increase in the antibiotic resistance, and the choices for treatment of cholera, the choice of antibiotics has steadily gone down. Okay. And for example, in the cartoon below gives you the scenario for other bacteria also. So the situation is not very pleasing. It's very gloomy. And for example, by 2050, people are telling that roughly 50 million people will die of the drug resistance. So it will be a huge toll. It will be a huge pandemic, a greater pandemic than the COVID-19 that we have seen, when people start die of the antibiotic failures. The infections will never heal. The fractures will never repair. The surgeries will never become successful. Simple operations like cataract and all will become so complicated that people will lose their eyesight. And then if the cataract is complicated, the infection may reach to the brain through the optic nerve. So like that is the scenario, and the things are being reported right now. Why wait until 2050? The statistics are horrible. What is happening basically is the sheer propensity of these organisms to import the genetic elements over a period of time. Okay, in the mixed infection settings, or when the water is contaminated, or when the handlers contaminated, when the food is contaminated, when the saline solutions are contaminated, when the rinse is contaminated, when the gloves are contaminated. So this kind of a scenario of horizontal gene transfer takes place, and there are quickly the virulence factors that are required. So the E. coli is so simple organism, very simple. You can cultivate. Why we work on E. coli is because it's very easy. If you want to work on a bacterial system, think of E. coli. In 2.5 hours, you get the turbidity that is required for you to work. Just inoculate your bacteria, go for lunch, come back, you'll get the turbidity, you can start working. Some of the bacteria like Mycobacterium avium, subspecies paratuberculosis, which we used to work earlier in good olden days, takes 6 months for the colonies to grow. How you'll do your growth curve studies, how will you do your functional assays, and how will you do your protein expression studies? You will never be able to do that. And that is how we use E. coli as our workhorse. And you see, E. coli is so beautiful. If you look at it, the adornment of itself with so many virulence factors. And then these virulence factors are actually supplemented, fortified, augmented, substituted whenever there is another bacterium or another community in the vicinity. So quickly with horizontal gene transfer, they achieve the maximum capacity to inflict maximum damage. And E. coli grows very fast. It overwhelms all other bacteria. For example, mastitis in cattle, the infection of the mammary gland, where, for example, there are other players like Staph aureus, streptococci, other gram-negative bacteria. But then when E. coli gets in there, it overwhelms all other bacteria and overgrows. And like that is the scenario of any other clinical sample. You see the E. coli is the first one to be isolated. And that is how it's better to study E. coli. And if we are able to control E. coli, we will be able to handle 70% of the infection burden. It is such a nasty organism. Although its genomic organization appears very beautiful in terms of virulence factors. But then it is not only one E. coli, it's not one lineage. There are several lineages of E. coli that are important. For example, we recently did the lineage analysis of E. coli in our paper. 2 or 3 of our papers that were published, E. coli and Klebsiella pneumoniae. And you see some of the super bug type of lineages like the ST131. You see on the left-hand side of the cartoon here, and there the brown color cluster is the ST131. These are the clonal lineages that have very high propensity to acquire drug resistance and become superbugs of a different kind. So these lineages are important. When we analyze, we found that several lineages of E. coli are there in the industrialized countries where these kind of infections occur. And they are also seen in our country, for example. And ST131 is one of the critical lineage with that. If we understand these lineages, it is important for us to understand exactly what is the genetic makeup of these organisms and which way they evolve. So we should never think that E. coli is one community. There are different lineages have different propensities, different virulence potentials, and so on and so forth. But then it is equally also important to identify certain targets. You see, then if you talk about only data, if you talk about how damaging it is, but then if you do not talk about whether there is any intervention that is going to come out. But the interventions, they are not like every day that you cannot find them. I mean, the discovery path is very difficult, particularly in this country, or particularly the anti-infectious discovery is very difficult. It is less rewarding. It is very time consuming. But then some of the things just they come by. For example, in Helicobacter, we saw this particular target 421. Okay. 421 of Helicobacter is the cholesterol alpha glycosyl transferase. We studied it in the context of drug resistance, whether these kind of things have anything to do with resistance. And so we found that when this particular protein is deleted from the genome of Helicobacter, the resistant bacteria become sensitive. When we put back this protein and substitute it, then the substituted recovered knockouts, they become resistant again. So that we found that this is very important because it's a component of cell wall biosynthesis system in Helicobacter pylori, and it will be pertinent to develop it as a drug target and see if it can be blocked. The resistant bacteria could be turned into sensitive and then treated. So pre-treat it with something that turns the resistant forms into sensitive forms and then treat with the regular antibiotics. That appears to be some of the solution in hand. Just as a passing example, I wanted to give. Other than that, we have also identified some of the inhibitors for this 421, and we have already published it. We didn't patent or something, so that anybody who has interest can develop a drug and try to help the Helicobacter pylori clinical community resolve the problems of antimicrobial resistance in Helicobacter. So just a passing reference, I wanted to tell you that this kind of treatments are also possible. This kind of interventions are possible, but that they take some time. There is an example also of aspergillomarasmine here on your screen, where the aspergillomarasmine A, when it is administered, it can just make the cell wall compromised, and then your regular antibiotic can work. I'm nearly coming to my end to that coming to an end of my talk. But before that, I would like to take you to or excite some nostalgia. Remember, the penicillin antibiotic is now getting there is now 100 years. We are getting 100 years, almost 100 years from the discovery of penicillin, and many people still consider that penicillin was a accidental discovery. Discoveries could be serendipitous. They may not be accidental. I see that there is a cause and there is a plan for everything to appear and take the stage. For example, during the World War, two important discoveries have taken place: the discovery of atom bomb and the discovery of penicillin. And you see, which has supported and helped the mankind? The penicillin has helped the mankind to save 200 million lives. How many million lives have lost by the use of atomic, forces of destruction? So many. So the discoveries of biological in nature, biological discoveries of antibiotics, so these things are so humble discoveries. But this discovery was not accidental. what Alexander Fleming has told in his memoirs. I would like to read for you that we all know that chance, fortune, fate, or destiny, call it what you will, has played considerable part in many of the great discoveries. The outstretched hand of opportunity I could see, he says. So what is that outstretched hand of opportunity? Could that be the hand of God that he has seen in discovering antibiotic penicillin? So people say that it was an accidental discovery. So let us dissect it 1 or 2 minutes if you had. Serendipity is not serendipity. It's a sagacity plus n number of favorable accidents that leading to very positive outcome. Something like you are winning the lotteries every time. Is it possible? Does probability support it? So high valued discovery is resulting in every accident. For example, Alexander Fleming left for his holidays and left his window open. And how is it possible for a seasoned scientist to leave his window open and left also a culture plate on the table? And you know what happened out of the 300 plus species of penicillium species, one moves to his building, then travels to his floor, then travels to his office and goes onto the plate where the Staphylococcus aureus bacteria were streaked already. And then this mold grows there. Alexander Fleming comes back and discovers that this mold has killed the bacteria. You know, I analyzed the weather data of Great Britain during that time. It was the September of 1928, and it was the warmest month in decades. So given all these favorable accidents, I consider that it was not an accidental discovery. It was as if somebody wanted that this panacea should be handed over to the human beings. Because otherwise the World War II would have amputated millions of arms, millions of people would have died. You know, just 20, 20 years before World War II, this discovery was handed and the doses were ready during that time. Otherwise, you know, 80 million is the official mortality of the World War. It would have been so much huge that the even humanity would have struggled to survive to this day. And that is how the discovery of penicillin, the probability doesn't support that it was an accident. It was a description, you know, it is not, there appears to be a supernatural outstretched hand. So that means it was a miracle that has saved 200 million lives. But now you see the data today on your screen. It was published in the Lancet and the data are horrendous. I mean, the every class of antibiotics and every given microorganism, if you map with the every given community and you see the incidence data, we see that we are now losing the positive effect we got of the penicillin and other antibiotics, and we are back to the square one. So now we are full circle of 100 years and antimicrobial resistance is going to take 200 lives back by 2040. So is there again a miracle that is going to take place? So I quote that God will not change the condition of a people until they change what is in themselves. So people will have to change their practices. That as to how they approach, you know, sanitation and hygiene as a main paradigm, how they curtail the development of such nasty bacteria around them and in their communities, in their food and drinking water, and how they save themselves of these infections and make others' lives also safer. Thank you so much. I would like to acknowledge all PhD and master's students of our lab groups at University of Hyderabad and in ICDDRB, Bangladesh, where I used to work. The University of Hyderabad and its Institution of Eminence grant, ICDDRB and its facilities, the Indo-German grant that we availed called 1673, and grant from the Indian Council of Medical Research, 2 grants, one just completed, another is running. These are some of the things that I would like to acknowledge. Thank you so much for your patient listening, and I would like to listen to your questions.
Yeah. Questions? Maybe we can. Yeah.
Hi, sir.
Yes.
Yeah, this is Deepa. So, like, it's a doubt. So, can we relate AMR with biowar?
You can see, for the purpose of relating it, you can relate it, but AMR is something that is very intricate. You know, it's very mechanized process, and biological warfare needs you. You know, people are already people have used in the World War the cholera bacteria and others. So toxins are very fashionable there, and as long as toxins are not affected by you know, evolution of bacteria, the people will still be happy who want to use them. But other than that, you know, the virulence may not be as such that damaging. You know, for example, if you augment the virulence of a pathogen by 20 times, 10 times, you know, that may not be as effective as a toxin, for example, which may justly kill millions of people through the water contamination or something. So theoretically, yes, but not very efficient. But we see that AMR is a public health problem at hand rather than a bio problem. So if I was to see it, I will see it from a point of public health.
Hello, sir.
Yes.
Sir, we came to know that mycobacterium, like bacteria, they lose their genome and thus they become specialist from generalists.
Yeah.
During this transition, keeping aside the possibility of AMR, do they become more virulent? Is there any relation to virulence?
Yes. So I mean there is an optimization of virulence. Okay. The parasitic phenotype itself is a virulent phenotype. Virulence is something that is damaging the host. So for example, if something is so much dependent on the host that host is not able to ward it off, then it is more virulent, I will say. For example, if a mycobacterial antigen which elicits a lot of host response, and such a response that host is mounting the response in a you know, unsuccessful manner, that it is not able to clear it off. So I will see this as a virulence. You know, so virulence is a relative term. Some of the virulence factors could be also metabolically required for the pathogen, so it will retain it. It will not delete it. Okay, and they may serve the dual purposes. For example, the isocitrate dehydrogenase in Mycobacterium is also a you know, antigen. It elicits huge B cell responses, but it is also a metabolic enzyme. So virulence is a relative term. Fine tuning of virulence happens because an excessive burden of DNA replication is casted off. So whatever is the remaining resources that the bacteria has, it focuses on its survival within the host. So effective survival in the host itself is virulence, because it is very damaging in terms of granulomatous inflammation.
Yeah, sir, it's a doubt. So when you're telling about the cholera control, it was like cholera was getting resistant to the bacteria faced through a mechanism called quorum sensing. Can you just explain what is quorum sensing?
Yeah, so usually in a ecological setting, the cholera bacteria, they sense whether phage population is there or not. How much is the phage population? So the phage population, you see it is written here. The bacteriophages are the viruses that attack and kill the cholera bacteria in the aquatic environments where the bacteria circulate. So the cholera bacteria, when in large numbers, communicate with each other using a mechanism called quorum sensing mechanism. You know, quorum sensing is very established signaling mechanism among bacteria to defend themselves against the viruses. So there is a definite signal is given when to multiply and overwhelm the phages. That is what is the mechanism that I was discussing.
Sir, this is Krishna.
Yes, Krishna.
Sir, this rainy season has started in India, and like many of our big cities like Delhi and in Hyderabad also we can see flood, and everything is coming on the road. So wastewater treatment is so poor that can we study or do surveillance on this scenario and give some policy recommendation to the government so that we can reduce the burden of the diseases.
See,
What's your
water,
opinion?
seasonal water stagnation is definitely a problem, but it is not that great problem unless it mixes with the drinking water. So whenever something overflows and get into the drinking water, that is the problem starts there. But mere stagnation itself sometimes is very problematic. For example, for transmission like leptospirosis outbreaks are reported after stagnation. Chennai and some of the cities in Gujarat, they regularly report because the leptospira penetrates the moist skin. And when the school going children, they walk through these stagnated water and all there through their skin, the leptospira penetrates. Like that, there is a possibility of other contaminations and all. Definitely there could be, effective management of this. The storm drains, the particular, the drainage mechanisms are very important. So drainage should be, management of drainage should be very purposeful, like it should work effectively during stormy situations also, during normal situations also. And all the time the drinking water channels should be very separate from the human excreta channels or the garbage channels. They should never see each other. Then only we can avoid, foodborne, waterborne infections and other things. So that is a very effective way to downgrade the infection burden. So infection burden, if it is less, we have more developmental indicators and growth, much healthier population. The healthier the population, more productive outcomes will be there.
Hi, it's Aaron J. Great talk. So the antimicrobial discovery pipeline has been dry for a while, right? It continues to be dry with no antimicrobials being discovered. How feasible do you think would a vaccine approach be for resistant bacteria?
The situation at the moment is that anything is welcome, but the vaccines have their own problems. For example, we cannot have one vaccine for everything. We can target disease-wise. For example, in gram-negative itself, there are 20-30 members. So in E. coli itself, there is a huge diversity. I said so many lineages. Every lineage has so many virulence factors. Every virulence factor has so much of diversity. For example, antigenic variation, phase variation. So these are some of the baseline problems in vaccine development. So AMR is a integrated trait. AMR is a conglomerate of resistome. Resistome is a outcome of anthropogenic, bacterial, genetic, ecological, climatological factors. So it's a very complicated scenario, and I wonder, if it can be targeted directly by just a single vaccine. Vaccines are always going to be against the targets. The targets should be fixed in the evolution. If targets themselves, they keep evolving, then I doubt the vaccine will be successful.
Hello, sir.
Yes, please.
So as you said, in the World War time, penicillin, that means the drugs in general, they used to protect the sufferings of the people. Now the same thing now, just because of drug resistance, reverse it.
Yes.
Is there any alternative approach to reduce this? Somehow we need to reduce it. So any technology or any rational or random strategy, or through anyhow drugs never end, because in meantime the pathogens cope up with the pace with that of the discovery of drugs. But that means it is not an end. Drug itself is not a solution for the control of the disease. So we must think in an alternative way, different strategy where you can really reverse the same. Is there any strategy?
It cannot be one single strategy, but can be a mix of a kind of an integrated approach. For example, repurposing of the old drugs, as I told you earlier, that some of the old drugs like chloramphenicol could work, like a magic even today against typhoid. So we'll have to convince the manufacturers to start manufacturing it. We'll have to convince the doctors to start prescribing it. They may not like it because they may have not been taught, because for 10, 20 years it is out of fashion. So there are some of the repurposing could be one of the strategy. Another strategy could be reduce the infection burden. Another strategy could be effective stewardship and campaigning, effective outreach and those kind of things. So there's an integrated approach. The infection burden, reduction of infection burden due to sanitation and hygiene appears to be a very effective thing, rather than drugs and all. They are also to be there. So drugs are for those who are suffering. But those who are not suffering but silently taking the brunt of it, for them, these are some of the practices like outreach, stewardship, healthy food and nutrition, then making them aware of the hygienic practices and those kind of things. And then on the top of it, discovery and repurposing should not stop.
Hi, Ahmed.
Niyaz Ahmed.
So just to add to the previous question, I was just wondering if it should be an integrated approach. Researchers have already started approaching AMR through One Health, using One Health by studying through different disciplines. So what's your thought on it, and how do you think it can be effectively operationalized, considering all different drivers of AMR?
Yeah, that is how, for example, I was telling that discovery is just one part of it. Discovery is target based. Discovery can be just for one organism. When we talk about, AMR genes or resistome, we talk about all type of organisms that are there around the human communities. And so, as you rightly said, integrated approach is the only viable approach that we foresee. But then we can, at the same time, also cannot discount for discovery efforts or repurposing efforts or academic awareness and public outreach campaigns and stewardships, proper use of antibiotics, proper correlation or association of production versus usage data as to how much is the requirement, how much is produced, how much is used, and also see how much residues are going into the environment. For example, restricting the antibiotic discard into in the environment from the factories will itself do a great deal, because a lot of organisms get sanitized because of the residues. So residues are going into the food also, for example. Residues in the food, residues in the vegetables, residues in the water and the environment. So stopping the exposure is one part, controlling the infection is another part, and discovery and repurposing is another part. So this is an integrated approach. And definitely there is a very high level of academics into the backbone of this. I mean, it cannot be with hand-waving approach. It has to be with a thorough thorough academic and mechanistic approach. There should be a kind of a evidence-based approach for each of these components. And that is how I see that there is also a great requirement of collaboration and people from different disciplines talking to each other. Great requirement for the funding arrangements of that sort, and for brainstorming and well wishes on the top of it.
Hi, Professor Niyaz. This is Anuran.
Yes.
So I'm just thinking of this situation like 100 years into the future or something. Yeah, I'm thinking of the situation about, let's say, 100 years into the future. So we have a limited repertoire of antibacterial drugs, right? Antibiotics, sorry. So at some point, since these microbes are constantly evolving and increasing their repertoire to tackle these antibiotics, at some point we are fated to lose, right? At some point we won't have any drugs left. So it sort of feels like hygiene is the way to go. And so you mentioned this particular thing that chloramphenicol lost, some of the bacteria lost its ability to defend itself against chloramphenicol, like something like a time scale of 10 years. So it feels like if you continuously use these antibiotics, then all the bacteria will sort of get all the genes necessary to ward off all antibiotics. But if you don't really use the antibiotics for, let's say, 20 years, 25 years, and then the bacteria will sort of like the microbes will go back to their stage where they'll be sensitive again. So it feels like if we allow civilization to not use antibiotic for 10 years, 25 years, like suffer, and then get it back in, and then use it in a very more like way more like systematic, controlled way, then it might that's like the only way I can think of, because this is a battle where there's no
Yeah, I
No,
agree.
I really want to know your
No, I
view,
agree
like,
with you. The thing is, you consider sulfur drugs, antibiotics, and all are around only for 150 years. But the humanity is not 150-year-old. We have been there, for example, sweet at least 10,000 years old that the evolved communities are living. So they have lived, they have thrived, they have migrated to different continents, they have built the cities, they have built the economies, they have built the kingdoms and dukedoms, and so many things have happened. So antibiotics are around only for the last 150 years, but the humanity has been survived for millennia. So
with the
that means there
diseases,
has
right?
been some survival system embedded into our societies, that we could resist all the things like plague came, the pox came, many different calamities have fallen on the human societies, they have survived. So we'll have to learn how to survive, in the absence of these magic bullets. And some policies, for example, from the government sides, for example, only when it is needed in the dire situations. For example, if somebody is dying and needs a shot of antibiotic, then only it has to be approved. But you see, otherwise, if we adopt a policy of blanket ban on antibiotics, then there will be a huge problem, because
Right.
people will die. So, but then other ways of restricting it, for example, giving it to animal in their ration for fattening, putting it into the fish ponds, in the poultry ration. So those things are to be controlled. As a veterinary student, I was taught that antibiotics have an anabolic effect on animals. Like they are the growth promoters and accelerators. I had a poultry nutrition and animal nutrition subjects, and in that it was taught that I'm not talking about Stone Age. I am talking about 30 years from now. So I was taught that antibiotics are good for giving into the ration, because they promote the weight gain by the animal. And that is continuing even today, that the animals are being fed in ration with antibiotics for fattening purposes, for achieving the growth of the animals, and then as a result, residues are being passed on into the eggs, into the meat, into the milk. So at least this kind of industrial scale use of antibiotics should be stopped. So no permission should be given at the point of selling for selling antibiotic as a powder or as a container to these companies. As a blister pack, as a capsule, as a tablet is fine. That is as a minuscule scale. But at a high scale, like in fisheries, in poultry, in domestic animal farming, there is a huge use of antibiotics. So at least our people should stop that. And see, it is religiously stopped, not on paper.
All right, thanks.
Yeah.
Hi, sir.
Yes.
So your one way of approach is to eradicate this antimicrobial bacteria. So is to have a hygienic life, and a hygiene means a controlled sanitary. It will eradicate this anti-resistant microbes, right? So for a time being, just we need a therapy or a medication side or a right. So as I've mentioned, I've seen watching this quorum sensing, which is resistant to the virus. So there is a one type of therapy like a phage therapy for a bacteria which are very antibiotic resistant. So is there any method we can inhibit that quorum sensing and we can promote the phage therapy? And my another question is, can we use a peptides rather than any large drugs which can easily be can be very efficient against these bacteria, rather than using a complex drugs Can we use a small peptides so that we can control this anti-resistant drugs?
No, I really appreciate your ideas. The thing is that the simpler we go, the smarter the bacteria move. You know, the simple things are very generic. Complex things are very specific. If we come with a very generic solution, they quickly adapt to it and develop a resistance. Phages also, for example, are very specifically tailored to a particular bacterial host population. So quickly, there could be a possibility that a capsular antigen evolves and the phage is not able to sit on the bacteria over a period of time. But nevertheless, there are many ideas. You know, some peptides are very effective. Small molecules are very effective. Some phages are very effective. So there is no doubt that these are some of the very nice alternative ideas. But then we'll have to think about, you know, some very generic approaches that appeal to the masses, you know, and in general, on a whole. For example, if we achieve sanitary, you know, we achieve, you know, clean drinking water, for example, 100% hand hygiene. For example, if we introduce hand washing in school children at the time of midday meal, for example, you can bring down student absentees by 75% because of the diarrheagenic episodes. Just what you have done is you have introduced a soap solution and water for hand washing and made it compulsory for every child before he or she sits for meal, just wash your hands with the soap solution. So with this, you can see that if granulated data are to be pulled, that effectively the infection burden could be curtailed. So the only you know, silver line of it is that we can control the infection burden, but we cannot control the infection when it has escalated, for example, and gone into the ICU and all. But those are only the minuscule of the cases.
Yeah, hi,
Thanks.
sir. Last one.
Yes, please.
Our last question. So I just want to know your comments on how the prevalence of horizontal gene transfer affect the spread of antimicrobial resistance.
Horizontal gene transfer is the major culprit in propagation of the resistance genes. So for example, how the genes are moving from one organism to other organism, one population to the other population, the major modus operandi is through the horizontal gene transfer. If this was not existing as a mechanism, then the problem would have not existed. This sheer ability of bacteria to integrate the DNA into their genome in itself is the main culprit. So maybe the mechanisms at recombinational level, if they could be targeted, you know, for example, but they will be global targeting. So all other, anything that can be acquired by them will be blocked. That will also affect any other favorable mechanisms also.
Hello, sir.
Yes, please.
I have one question in regards to, so have you studied this mega slums that are in India? Like, for example, if you take Dharavi, and if you compare it with the refugees, basically. So Dharavi is a slum wherein a lot of folks travel throughout the city. Everyone's traveling everywhere and they are mingling with a lot of people around as well. And wherein in refugees, a lot of movement is restricted. So would you see complex, more resistant pathogens here compared to there?
Dharavi is a very developed urban slum, I'll say. It has quite good facilities. You see drinking water available of the provisional stores, food, everything. So it is very well developing. And we do not have comparable refugee populations within our city, our country. So since it was a country setting of a particular country where simultaneously the slums as well as refugee populations were available, and simultaneously these things were there, we compared and studied justly because they were also accessible. Given the opportunity, definitely will visit and study every urban slum where there is a ghettoization or high population density, mismanagement of the human excreta and the clean drinking water. That will constitute a very ideal site. And whenever we use sample and subject it to global sequencing and all, you'll definitely find new genotypes, new combinations all the time. It will be crucial to know as to from where they have come, who introduced them.
One more thing, sir. So we, as an undergrad project, what we did was we collected these juices that are available that are managed by railway authorities, and they are available to they sell them at the railway stations. We did an E. coli culture for them, and we found a lot of them having E. coli. And this are being drunk, like a lot of people drink it, like more than 1,000 people, more than that actually. So like what, like I don't understand, like what role does railway authorities play here, or do they not check all of this at all? Because they manage it a lot.
This was your project, undergrad project?
Yeah, like we just tried little bit.
Yeah, so food safety is a very general problem, and it has to be addressed from that domain itself. And if we do that there's an organized sector, there is an unorganized sector in the food supply. There are fast food, packaged foods, different kind of foods. So these sectors have definitely some problems, and that is how, realizing these problems, the government of India has set up the FSSAI, Food Safety and Standards Authority of India, and it has been mandated, to exercise global standards, so that the food that is, the packaged food or ready to use food or whatever food that is being vended to the communities has some standards. So I am sure with their efforts and their stringent practices, we will not be able to see the situation that you are describing.
Thank you, Dr. Niyaz, for this fantastic talk. And also thanks for orienting us to the reality of pathogen evolution and linking that to the environment. Thank you so much.
Yes, thank you.